What's dark life? It's a term for an entire class of microorganisms that have been invisible to scientists for most of history. But with newly available technology, we're finally starting to discover them. What makes these organisms so valuable is that increasingly, people are turning to bioremediation for cleaning up toxic chemicals. And the more bacteria we find, the better chance we have of finding what we need. But why has so much of life on Earth been hidden? To answer that question, we have to understand how biologists have traditionally studied bacteria.
What we do is we take bacteria from the environment, we'll streak them on a plate and let them grow, and whatever appears, we can separate them out into their individual selves and study them. But what that does is we can only study the organisms, the bacteria that grow on these plates. And so if we haven't provided everything they need, they'll be invisible to us. They will seem not to exist.
When we go into an environment, we tend to collect all of the organisms that are present and extract their total DNA and sequence it, and that's called metagenomics, so total community DNA sequencing.
Jill Banfield at Berkeley and Laura Hugg, they went to extreme environments. They sampled like 8 different kinds of environments, and they ranged from thermal vents, and then they took swabs from dolphins' mouths.
All that collected DNA is shotgun sequenced and analyzed using supercomputers like the NSF-funded Comet at SDSC. By looking at changes that build on top of each other, we can determine which organisms are variants of existing species and which are completely new. This is called sequence alignment.
Now we're getting thousands and thousands of DNA sequences from creatures we've never seen before. Things that were basically, it's referred to as dark life. Life that we had no idea existed because we had no way of detecting it.
So why is it important to discover these new life forms? For Dr. Hugg, her work in metagenomics dovetails with her work in bioremediation, or the use of bacteria to break down contaminants.
The environments that I'm interested in right now are municipal landfills, and so they're very heterogeneous, they're multiply contaminated, they're not very glamorous, but they are a really good place to look for previously unknown or understudied lineages and for organisms that have adapted to a contaminated environment. The site that we're working with actually traps the methane that's emitted from the site, converts it to biogas, and powers both their site and feeds some power back into the grid. I have a student who is looking at how bioplastics are degraded, whether or not it would be advantageous to develop something like a pre-treatment spray of microorganisms that one could conceive of spraying over garbage as it is being compacted and put into a landfill. And that would potentially speed up the plastic degradation. I really want us to get ahead of the curve on that, to have tools in place for when we realize that actually really this is a problem, this is threatening groundwater supplies, this is threatening drinking water supplies, this is more carcinogenic than we understood it to be, or just, we really should perhaps clean up the mess that we've been making.
Dr. Hugg's work in uncovering dark life is a key example of how abstract technologies like computational genomics can have a real impact on our quality of life. We're just scratching the surface of what's possible when we work with nature instead of against it. And bringing bacterial life out of the dark is just the beginning.